We are recording today. So, we are on Module 2B. All right, so the second half of 2, that means next week you will have a quiz over Module 2, and I'll need you to bring your study guide, all right, for that, and then you'll sit down and take your quiz, show me your study guide, and we'll take care of that, okay? All right. So, we started our experiment already. It looks like that. Does it look like I have something over it? Like a saran wrap or something? Maybe not. Okay. I don't think our book asks us to do that. Is that just a, y'all see what I'm saying? It looks like it has something over it. But here it doesn't. It looks okay. All right. All right, I put that picture in for me so I remember how to set the thing up. All right, it explains it in the book and everything. It's all in your book. In fact, you'll be looking through that, reading through that to get your conclusions and stuff when it comes to writing up that lab. All right, so we're going to start today with homeostasis, okay? Keep calm and maintain homeostasis. What does it mean? It's the tendency of living organisms to control or regulate changes in their internal environment. All right, it's all living things are trying really hard to not be overly stressed. We don't want to be stressed out. And when we get stressed out, we do something to counterbalance that. All right? In other words, the ability to survive in the midst of these environmental stresses. Y'all understand stress, but here we're talking pretty much about physical stress. For instance, if you were cold, that would be a stress on your body, and your body would do something to counteract that stress. You would start to shiver. Yes, we talked about that. And if you were hot, then you would do something to counteract the heat. You would sweat. All right. So here we are looking at organisms in the marine environment, and here's the big question. How in the world do these guys survive in the salty sea? Salt is a major stressor, okay? So here we are. We're looking at stress. Salt tends to inhibit life. Do you know this? I probably shouldn't tell you this, but you know it kills grass. If you sprinkle, bless you. If you sprinkle salt on grass, it will kill it because it's so caustic to it, right? We used to, well, if you can write a message in someone's lawn with salt. It'll take a while for it to manifest, but don't do that. It used to be something we joked about doing. We never actually did it. So, but you could, you know, if you wrote a message, it would die and you could read it, you know, that kind of thing. So here we're looking at some salt. So this is the Dead Sea over in the Holy Land, right? And there's some sort of rope or something right here. And the Dead Sea is dead because it's so salty. All right, it's really the lowest place on the earth that's not underwater. So they call it the belly button of the world. It's like everything ends there, you know. Interestingly, it's the Dead Sea. There are some people who believe that that's where the Garden of Eden was, which was life, right? And so we don't know. I will tell you it's very, very clear. You can see to the bottom of it because there's nothing growing in it. The stuff that makes our, you know, water murky and stuff is the creatures swimming around in there. Your pond water and stuff, there's creatures. No creatures here because it's just too salty. Now, we have a salt lake in America called the Great Salt Lake, and it's not as salty as this. And so there are some creatures that actually live in the Great Salt Lake, and they're like little krill type creatures. They're very small little shrimpy things, and they make the place stink. It just stinks. But this place is not. You can float in it really easily and stuff like that. But what's happened on this rope or whatever this is that the water, the salt has, when the water got on the rope, the water evaporated and left the salt behind. All right, it leaves this giant, like almost sculpture there, and all this salt right here. I had a friend who went and floated in the Dead Sea, and she said it felt kind of slimy. I thought that was interesting. You know, so it'd be fun to go see that and float in the water. One thing we just have to remember really quickly what an ion is. For those of you taking chemistry, you should remember that. If you haven't taken chemistry, just remember it's an atom that's lost or gained an electron, and salt is really, really good at that. Chemistry kids, we know that. We did that today. Where we took sodium chloride and made it to the ions that are floating freely. So when you make salt water, for instance, the ions are freely floating in the water like that. So remember that these ions, therefore, will act a little differently toward living creatures. All right, our first definition here, diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. This is if somebody is cooking popcorn in the kitchen, in the microwave, right? Pretty soon all of us are going to know it. We'll smell it and we'll say, oh, somebody's making popcorn, it smells so good. And then if they burn it, we know it then and we think, oh, that's horrible, right? They messed it up. Because you can smell it. So that's just diffusion. Those are those atoms of popcorn smell coming to every bit of the building. We're pretty far from the kitchen. I don't know if it would make it all the way here. I definitely have bubbles in here, so that's good in our experiment. Okay, so it turns out that living things have a plasma membrane. The cells have a plasma membrane on it. We're going to do a little lab on that today in a little bit. It allows small molecules to pass through in diffusion. So diffusion works really well when we're talking about small things like water. Water is H2O. It's very small. Sodium and chloride are very small. They can typically go through a plasma membrane because they're small enough. Now osmosis is a little different. Osmosis is the diffusion of water across a selectively permeable membrane. This should all sound familiar from Biology 1. All right, so a selectively permeable membrane means it lets some things in and other things it keeps out. So water is small enough, it generally moves from an area of low concentration to high concentration under the dictates of diffusion. It does that to help equalize the concentration. But keep in mind, we're talking about salt water here. These creatures are all living in salt water. So they have to come up with strategies to deal with this salt water that's all around them. One of them, of course, is design. They've been designed to live in salt water. Okay. We have a little video, just as a reminder. This is from Biology 1.
Diffusion tends to even out concentration across a permeable membrane. In this beaker, we have a high concentration of solute to the right of the membrane and a low concentration to the left. Since the solute is free to travel across the membrane, the random motion of the solute molecules works to even out the concentration, so that by the end, the concentration of solute is the same on both sides of the membrane. Osmosis also evens out concentration, but in a different way. Once again, we have a beaker with a high concentration of solute to the right of the membrane and a low concentration of solute to the left. This time, however, the membrane is semi-permeable. It allows the solvent to travel across, but not the solute. The only way the concentration can be evened out then is for the solvent to travel from the area of low concentration to the area of high concentration. This dilutes the solution on the right side of the membrane, and it concentrates the solution on the left side until the two solutions end up having equal concentrations. Diffusion and osmosis then end up doing the same thing. They equalize concentration. Diffusion does it by the solute traveling across the membrane so that both sides end up at the same concentration, whereas osmosis does it by the solvent traveling across the membrane.
All right, so really that semi-permeable membrane, that selectively permeable membrane, is really allowing the water to go through. Other small things can go through as well. Think of it like a screen on a window, right? The bugs can't get in because the screen is too small, but air gets in because it's small. Okay, so what is selective permeability? It is the plasma membrane in cells that allows for only selected molecules to pass through. And depending on what organism we're talking about, those might be different things. For humans, of course, our selectively permeable membrane will let water in all day long, but some things like potassium and sodium, it has to use a pump to get those in and out. You may remember that from Biology 1, the sodium-potassium pump. It has to use active transport to do that. I have this little thing here that shows how the water's going to pass through that orange membrane thing freely, but those big giant things on this side can't. They're too big to pass through. And that keeps that solute over there. Here's another picture showing it. The water are the little purple things, and then those giant deals, that's sucrose. Sucrose is table sugar, so sugar can't pass through. It's just too big. So disaccharide, very large. All right, this is an experiment that's in your book that you may do at home if you like for extra credit. Take a picture and show it to me. That'll be good. What you do is you take 2 eggs and you put those eggs in vinegar overnight, and what'll happen is the shell will be gone the next day, and you'll just have the membrane around the egg. The shell's completely gone. We call that the rubber egg, right? You take one of those eggs and put it into regular water, and the other one into high fructose corn syrup, like Karo syrup, and you'll see this diffusion thing that goes on. It takes time to work, and so therefore we don't do it in class because it won't work very well. But it does say distilled water, because if you use like a salt filter or salt softener or something, that will affect the amount of water that'll go in, because there'll be salt in it. So distilled water, same thing as RO, reverse osmosis water, would work. Most bottled water has minerals in it. Okay, so that would be extra credit if you wanted to, and tell us what happened. You tell me, just take a picture, tell me what happened. Okay, so we're gonna, don't write these words down just yet. Okay, I've got a better definition for you right here. We're gonna look at, here are 2 little fish right here. I think it's the same kind of fish. They're both salmon, I think, yeah? Does it tell us that? And we're gonna look at coping mechanisms for living in high salt water, okay? So we have this one group called osmoconformers, and that means they're gonna have their osmolality, that's gonna be their concentration of salts in their tissues, is gonna conform to the environment. And then the osmoregulators, or osmolality, is going to be regulated by some process inside their body. All right, so you can kind of think of it, we're gonna get to temperature in a minute, which we'll be looking at cold-blooded versus warm-blooded, so kind of think of it that way. Have you ever heard that if you drink salt water, it'll just make things worse for you? It's true, don't ever drink salt water. And I'm talking about if you're in the ocean on a life raft and you're dying and you say, I'm so thirsty, there's water, I'm just gonna, no, you'll die quicker, okay? Because of the salt will draw more water out of you. Okay, so that's the problem here. We have animals that are struggling with the possibility of becoming dehydrated. And we all know dehydration is bad for us. All right, so here's what we're gonna write down. An organism that has the same concentration of salts internally as the water externally. Okay, and sometimes the salts are different. Sometimes the salts in their body are different from the salts they're swimming in. So we might have sodium in the water and they might have magnesium in their body, or potassium or something like that. So the quantity of dissolved salts matches internally and externally, even though the type might differ. So these are conformers. We might think of these creatures as being too lazy to try to fix things in their body, right? I don't know if that's rude to them, just call them lazy. Here's one of them right here, a starfish. Starfish just says, yeah, this is where I am, this is what I'm gonna be like on the inside. They just change them, they just conform, that is, to the outside. They change for that. Another guy, what is that?
Lobster.
Yeah, lobster does that. An osmoconformer. So you can also maybe think of these as less sophisticated because they don't have that internal mechanism to change. Here's our osmoregulator. That's an organism that regulates its internal concentration of salts. That's our osmolarity and osmolality. So bony fish, those are examples here. We'll look at some specialized fish here in a minute, the salmon in particular. So it has a mechanism to regulate this, to get rid of some of the salt. Okay, we'll be looking at birds and things and reptiles later in the year and seeing how they deal with salt, you know, because some of the ocean birds actually drink salt water, you know, birds. And they have to be able to figure out how to deal with that salt.
Do you need to write down the term osmolarity?
If you want to, you can. It'll probably bring it up in the book. It's just really talking about that osmosis, you know, gradient kind of a thing. Where is it more salty kind of a deal. All right, are we good? So what they have is they have these fancy cells called chloride cells that are actually in their gills. Okay, so we're talking about bony fish right now. The gills contain these specialized cells right here, and they are very, very rich in mitochondria. Remember what mitochondria are for? Powerhouse of the cell, never forget that, right? So the mitochondria are making ATP, making adenosine triphosphate, making energy. All right, so what we have here, they're going to use energy, in other words, to deal with that chlorine, that negative ion that's coming there. So our sodium is a positive ion, our chlorine's a negative ion. So we're going to have an ion exchange, leading to osmoregulation. It says here metal exchange. The metal is the sodium. Sodium's a metal. It's just a technical term for some of the elements on the chart, right? So here is our creature, right? Here's our fish. And you know, fish as they're swimming, they will gulp water. They actually drink water, for one, but they push water through their gills for a couple of reasons. We already know one of them. They push it past so they can get the oxygen out of it. That's what their gills are for. Their gills are their respiratory organ. So they're breathing that way, all right? But also they have specialized cells embedded in there that deal with the chlorine by using energy to negate that ion that's in there. All right, so they're able to then therefore change that in their body. It's quite fascinating. So they use chloride cells. Okay, here's our salmon. Now salmon are very strange. We call these creatures anadromous. And anadromous means they live part of their life in the salt water and part of their life in the fresh water. So think about it. If I took a saltwater fish, they're very expensive, by the way. If you go to a pet store and want to buy saltwater fish, they're expensive and they're beautiful. If you've been to the dentist up here, there's beautiful fish swimming in that tank because it's a saltwater tank. Freshwater tanks, the fish are just kind of dull. They're just not as pretty. They don't have those bright, yellows and blues and that kind of thing. But if I took a saltwater fish and brought him home to my regular aquarium and put him in there, it would die. And it would die because more water would come into its cells than are supposed to come in there and he would explode. Now, it wouldn't be very dramatic. He would just go, kind of puff up a little bit and die. If I had a freshwater fish and put it in my saltwater tank, he would shrivel up and die. All right, so it's super important that they know how to deal with the water around them. So what in the world is the salmon doing? It's going from salt water to fresh water, back to salt water. Oh my goodness, it's crazy. And so what they do is they have this mechanism. When they are in salt water, they increase the amount. They actually drink a lot. So not just pushing it past their gills, but drinking it into their stomachs. And they have very decreased urine output, so the urine is very concentrated. When they're in the fresh water, they decrease their drinking of water, and they increase their urine output, so very dilute by more water going into their urine. All right, so see, this is that picture we had earlier, right? So you can see here, these are both, I guess, different kinds of salmon. And so you can see here in the, is that fresh on the top? Drinks little water. Trying to see which one's which.
Freshwater's on top.
Freshwater's on top. Okay, thank you. All right, and so you can see the drinks little, drinks a lot. So then it can get rid of it through its little fancy cells, those little chlorine cells right there. All right? So that's how they deal with it. Not all fish do that. Most fish want to live in one place or the other, either salt water or in fresh water. But there are these anadromous types that do have this special coping mechanism, and you'll read about those guys this week. All right, temperature's another one. As a general rule, metabolic processes slow down the lower the temperature. Y'all know this, right? When it's cold, we don't want to do anything. We just want to sit under a blanket and watch the ice outside, like last year, right?
Should we write the first one down?
I think they want you to know this. For every 10 degrees Celsius increase, the metabolic rate doubles.
Never
Yeah.
erase this.
So here's what you just want to know. Warmer temperatures are for more active fish. Colder temperatures are less active fish. And there's another experiment in your book that I just can't bring myself to do. And the reason is you have to get a live fish or 2 live fish. If anybody has fish and would like to bring them and show us this experiment, we can do that. But my problem is I don't know what I'm going to do with the fish. If I bought some fish, one of my students came to class last week with a little betta fish in a cup, and I was like, well, I could do that. But then I'd have a fish. What am I going to do with it then? I'm not big enough to eat. Yeah, I would have maybe donate. I know. My third graders, they talked about goats, and she said she had goats. I said to eat, and they were horrified. They said, well, no, we use the milk to make soap. I said, oh, well, people eat goats.
Betta fish are also really fun to mess with. You just put a mirror up against their.
They fight?
They ram into the glass and it.
Yeah, see, that's the, I would have 2 betta fish, and they can't put them together.
We had to fish.
They'll kill each other.
We had to fish set a fish when we were kids, and so my sister and we messed with the betta fish a whole
Okay.
bunch. And it was a lot of fun.
Psychological torture.
No, we didn't die.
That's surprising. We had a cardinal in our backyard who did the same thing. Because birds can't see glass. So it just
I had
saw
one
its
in my
reflection
window
in
who
it.
just would hit
over
my window.
and over again. And at one point, I think it knocked itself out. We were like,
I kept putting stuff up so it wouldn't reflect as much, but it still kept
Oh.
slamming into it. Okay, so the experiment that's in your book is you actually take your little fish and you make the water cold for one of them, and you make the water warm for the other one, and you watch how many times they go whoop, whoop, like that. You know, make their little mouth go whoop, whoop, and you count it. And I guess I'll just get y'all to just trust me that when in the cold water they do less. In the warmer water they speed up. Woohoo! Okay, and they probably swim around. I don't know. Anyway, I think the book says goldfish. Have y'all heard about goldfish? People get goldfish, right?
Yeah.
And then they get tired of feeding and taking care of the goldfish. They throw them in a pond, and then they get like this. They get big. And then other fish are getting eaten by these fish, and so the fishermen are like, they catch a giant goldfish. But then one fisherman did say, they're kind of tasty. So I was like, all right, well, you can eat them. Then there you go. Livestock, right? There you go.
more than just a cracker.
Okay, now of the stuff you're going to learn about in the second half of Module 2, this to me is the most complicated part of it. Okay, and it has to do with temperature. It's easy enough to say something's cold-blooded or something's warm-blooded, right? That's easy. But we're going to have other terms. You know how scientists are always trying to define things with new scientific-sounding terms? We're going to have that today, okay? So when we're looking at an ectotherm, so that means cold-blooded. Okay, that's kind of the easy way to say it. Y'all know you know what I mean when I say cold-blooded, right? This is the one, sort of like our osmoconformer, right? It's just going to be whatever the environment is. Their body temperature is going to be whatever the environment. So for instance, right now the temperature in this room is 72. If we were cold-blooded, our temperature would be 72. Is our temperature 72? No, it's 98. Our temperature is 98.6. It's almost 99, right? So we definitely regulate our temperature. We are not cold-blooded. We are warm-blooded. All right, but the technical term for a cold-blooded creature is an ectotherm, but the ectotherm can have two different kinds of manifestations. One that's a strict ectotherm, and one can be a poikilotherm. A poikilotherm. All right? So ectotherms' body temperature is controlled by their environment. We're going to see the difference between the poikilotherm and the ectotherm here in just a second. In fact, we're going to watch a video that's kind of long, and the guy has an accent, but after a few minutes, you get to understand it. He's got like a German accent, yeah. He's good, das ist gut. All right, so body temperature controlled by their environment. The easy way to say it is they're cold-blooded.
Why cold though?
Good question. What if they live in a warm place?
Yeah.
Yeah, it's just that they don't have a way to warm their bodies. So they're naturally cold, and they go as cold as it is, and they can't warm their bodies up. Unless they're a poikilotherm. And we'll see how they do that here in just a second. All right, has everybody got what they need for this slide? All right, so an endotherm is an organism whose internal body temperature is a result of internal sources of heat. In other words, a warm-blooded creature like humans. We have something in us that causes us to warm up. And what that is combustion. You know what combustion is? Burning. We're doing that right now. We are burning your breakfast. Right now you're burning your breakfast. All right, we're burning our food.
I was gonna say what
If you didn't eat breakfast and your body says, feed me, Seymour, please feed me. Because you've got to have something to burn so you can make your ATP. Okay, you've got to be able to make your energy, and that comes from the food you've eaten. If you don't eat anything, then you'll start to take your reserves, we call fat, and we'll burn that. All right, instead. So you're not going to drop your temperature because of lack of food. No, your body won't let you do that. It's going to go back to that homeostasis. I got to find my food somewhere. Let me find some fat. I'll take that and I'll burn it. And I'll make some ATP off of that. It's hard, it's a lot of work, but you can do it. All right, so in the marine world, the warm-blooded creatures are ones like that. Oh, that's a baby sea lion. Yeah, sea lions are really cute. Seals are frightening. There's that one movie that the seal, that cartoon, you know what I'm talking about? Happy Feet or one of those? It's like nightmare stuff.
I don't even remember that movie because I hated it so much.
Oh my goodness, yeah, all I remember is that seal. And
They have seals in San Francisco.
But
I've seen them.
actually they're sea lions.
Oh, are they?
Yeah, I've seen them too in the
Some of them are
out there,
fascinating.
and they sit up on the thing and look at you and go, huh, you know, they talk to you. You can tell because he's got little ear flaps right there, and he's cute. All right, he's cute. Also, this is another type of homeotherm or warm-blooded creature is a dolphin, right? Those are mammals. So what is homeotherm? Homeotherm is essentially a synonym for the warm-blooded, right? For the we have an organism that maintains its controlled internal temperature using its own heating and cooling mechanisms, therefore warm-blooded. Its internal body temperature does not change. So your body temperature does not change unless
We have a fever.
you're sick. And your body, that's part of homeostasis too. If you're sick, your temperature will go up because the rising temperature will make it an inhospitable place for a virus to survive, or an inhospitable place for bacteria to survive. In other words, fever is your friend. Fever's good for you. We spend a lot of time taking stuff to take our fever away. If it gets really high, I understand that. But if it's a low-grade fever, I just let it ride. Let it ride. Your body's doing that for you. It's a gift to you. All right, so now let's get to our poikilotherm. Poikilotherm. All right, so the poikilotherm is an organism whose body temperature changes with its surrounding environment even though it is cold-blooded. So it changes with its environment, that's right, that's cold-blooded, right? But, okay, sorry. That's the poikilotherm. An ectotherm temperature is controlled by its external environment. That's cold-blooded. A poikilotherm may have ways to make its temperature different than its environment. Okay, so we're going to watch a video that's going to help us here in a minute. A poikilotherm is a cold-blooded creature that can warm itself up a little bit through moving, okay, muscle movements. And that's how they do it. For instance, sharks, mini sharks, not all of them, mini sharks, tuna, these are large fish. They have very large muscles. Okay, so, you know, a tuna fish is as big as a table. You know, they're huge. And they have a lot of meat. That's why they're really good for harvesting for food. Because you can get a little can, lots of little cans out of that giant tuna right there. You know, we eat them usually in a can, right? They cannot completely control their temperature, so they're endotherms, but they're not homeotherms. They are not warm-blooded. They don't have an internal mechanism to do that. But they have a ability to move and warm up. That makes sense, right? If I move a lot, I'm going to warm up, yes? So they can do that too. Okay, so that's the big difference right here. Here's a tuna right here. Think of it as big as that table there. They move their large muscles to gain some warmth. All right, so this is the video I was explaining to you. It's a really good video. It's kind of long, and the guy has a very thick German accent. But at the end, we'll all be talking this German accent too, y'all. Das ist gut. So, and he will tell us about many, many things, including, if I remember right, the poikilotherms.
Animals typically use one of two strategies to regulate their body temperature. On the one hand, we have birds and mammals, endotherms or warm-blooded animals, capable of physiologically regulating the internal temperature within a narrow optimal range. On the other hand, we have ectotherms or cold-blooded animals. They cannot maintain a constant body temperature, so instead have to rely on environmental heat sources. As a result, their body temperatures fluctuate with the temperature of their environment. This group includes reptiles, amphibians, and fish, including most sharks. Because of the limitations of ectothermy, cold-blooded animals thrive particularly in warm environments and are most diverse there. Sharks, for instance, are most abundant in tropical and subtropical waters, where water temperatures are ideal to support a life as cold-blooded apex predators. Sharks that inhabit colder waters of higher latitudes or greater depth don't have this luxury and typically face substantial limitations. Sleeper sharks, a group of sharks that successfully inhabit the coldest regions of our oceans, have to live with a very slow metabolism to cope with the frigid temperatures. This not only limits their growth rate, but also their speed. They are one of the slowest swimming fish in the ocean. However, a small group of sharks has managed to overcome the limitations of living in colder waters without sacrificing speed, agility, and growth rate. They evolved in such a way that they can elevate and maintain body temperatures above the surrounding water temperature. temperature, effectively becoming endothermic. The most famous of them is the great white shark. Only 7 species of sharks, all of the order of mackerel sharks, as well as a few representatives from the tunas and billfish, possess this unique
This
ability.
just says warm-blooded.
Through certain metabolic processes, these fish can maintain body temperatures in certain parts of their bodies that are 5 to 14 degrees Celsius above the ambient water temperature. Think of it as a sort of regional endothermy. For great whites, this means that their flanks and viscera are warm, while the heart and the gills are cold. This is an evolutionary advantage that allows them to travel extensive distances into colder waters or dive to great depths, taking advantage of the rich feeding found there, while still being able to function effectively to capture even swift and agile prey like marine mammals. Additionally, it also heats their brain, reducing the risk of a sort of brain freeze and improving their eyesight when hunting in these cold waters. It may also allow for an increased rate of digestion, giving them faster access to the energy from their food. By holding their young in warm uteri, mackerel sharks might even enhance the development of their embryos, allowing for shorter gestation periods and higher birth rates. Researchers suspect that all this is a strong driving force behind the incredible success of great whites as a species. But how exactly do they achieve endothermy? As in humans, sharks have a closed circulatory system that supplies the muscles and other organs with oxygen and disposes carbon dioxide and other waste products. Unlike in humans, however, the gas exchange doesn't happen in lungs, but in gills. Furthermore, the gas exchange isn't part of a separate circuit that sends the oxygenated blood back into the heart first. Instead, the blood is sent directly through the body after passing through the gills. Here's where the systems of cold-blooded and warm-blooded sharks start to deviate. In most sharks, the red swimming muscles are located along their flanks, directly below the skin. The big dorsal aorta, a central running artery, supplies them with the necessary oxygen through a few arterial branches. Afterwards, the carbon dioxide-rich blood is pumped back towards the heart via the central vein. Most of the metabolic heat sharks produce is generated inside their muscles while swimming. But because water conducts heat 10 to 20 times better than air, this heat is normally immediately radiated from the skin and the gills and lost to the environment, resulting in no significant temperature increase. The great white, however, has evolved in such a way that it can greatly reduce this heat loss and thereby maintain an elevated body temperature. This is only possible through a series of modifications to the structure of their swimming muscles and the circulatory system. The first important change is that the red swimming muscles are buried deep within the shark's body, on each side of the backbone. Additionally, there's much more of it. In tunas, nearly 75% of the total weight is composed of these muscles. Red muscle, compared to the predominantly white muscle of cold-blooded fish, is much more powerful and produces more heat as a result. For that, however, the muscles require a much greater supply of oxygen. The additional oxygen is provided by enlarged arteries along the flanks of the fish that redirect blood inwards through a dense bundle of smaller arteries and veins. This fine network is called reed mirabile, which roughly translates to wonderful net. In some warm-blooded fish, additional retia are found branching off outwards from the dorsal aorta, but those are species-specific. So how do these changes trap heat inside the body? One factor is certainly that through the central arrangement of the swimming muscles, far less of the red muscle mass has direct contact with the water, which reduces the immediate heat loss. But much more important is the extensive countercurrent heat exchange, enabled by the dense array of veins that supply the muscles with oxygen. This heat exchange facilitates heat retention within the red muscle. But how does it work? Because of the countless small arteries and veins that pass very close to one another, carrying blood in opposite directions, the retia act as heat exchanger. Through this intimate countercurrent blood flow, most of the heat is transferred from the veins to the arteries and cycled back to the muscle that produced it originally. In contrast to concurrent flow systems, where the continued heat exchange between the two liquids will eventually lead to an equilibrium that is somewhat in the middle of the two original temperatures, in a countercurrent system, the exchanged heat is constantly carried away back into the muscle, pushed forth by new cold blood. This means the upper stream will always have the lower temperature, and more heat can be exchanged. Here, an equilibrium is only reached when nearly all the original temperature difference was exchanged. This makes countercurrent exchange so effective compared to concurrent exchange, as nearly all the metabolic heat in the venous blood is transferred to the arterial blood, thus conserving most of the muscle's temperature. For comparison, here is the cold-blooded fish again. The few arcs supporting the red swimming muscle in this setup aren't nearly dense and close enough to each other to allow for countercurrent heat exchange. And even if there were, it would only mean that the heat is directed back towards the flanks of the shark, where it would be quickly lost to the water. This is why this internal structure will never work in preserving heat. But countercurrent exchange is not only utilized within the muscle of warm-blooded fish, it is very common in nature and has a variety of uses. Its use isn't limited to heat either. It works just as well for gases and dissolved liquids. For instance, almost all fish utilize similar systems inside their gills to make the gas exchange with water as efficient as possible. Whales use it to minimize the heat loss through their tongues when filter feeding. A similar arrangement of whales prevents extensive heat loss through the legs of dogs and Arctic foxes. But countercurrent exchange isn't only used to stay warm. Sheep, for instance, have heat here inside their brains to prevent extensive body heat from entering the brain and causing cognitive damage when running for longer periods of time. It even helps desert animals to stay hydrated by reabsorbing as much water as possible from their urine. As you can see, countercurrent exchange has a multitude of applications in many different kinds of animals, not just in sharks. And in most of these cases, it is not only important, but vital to their success today. This still does leave one last question, however. Why, and more importantly, how did the ability to regulate and elevate the core body temperature evolve in these fish? And if the ability is so essential to their success, then why, in an ocean full of thousands of different species of fish, do only a handful of them have it?
So I would say design. That'd be my personal deal. So when he kept calling it a warm-blooded shark, really, it's a poikilotherm. Because how is it getting warm-blooded? It's getting warm-blooded from muscle movement. Okay, so that's how we're defining our poikilotherm. All right, so we have a poikilothermic. We have fish, reptiles, invertebrates. They're also considered ectothermic. Yeah. Okay. Our endothermic, though, they can be considered endothermic poikilotherms. Would be our large sharks, tunas, and billfishes. Those are the ones you go deep sea fishing for. All right, and our homeothermic, some deep ocean creatures will be ectothermic, and birds and mammals will be endothermic. Okay, so the homeothermic, it's doing something, right? Poikilothermic, it's doing something over here in the endothermic area. So we just have you have a little chart to fill in. Is that what you're doing? Okay, good. All right, so that's just a breakdown of how we can categorize these creatures. We have another video coming up pretty soon. So. All right, good. All right. So can an organism be a poikilotherm and endothermic? Yes, just like he was saying in the video there. Okay. Internal muscles warm the organism, but it may not have a way of maintaining that in the cold water unless it keeps moving. You heard about sharks always moving?
Yeah.
That's true, actually. They also don't float very well, so when they stop moving, they sink. Yeah. So, all right. This is going to show us the actual muscle of the shark.
The great white has a very high metabolism, and it needs to feed on rich, fatty prey such as dolphin, whale, and seal. However, this high-calorie prey often hides out in the colder, hostile waters, where most sharks can't survive. But the great white can increase its body temperature 18 degrees Fahrenheit higher than the surrounding water. The great white is not a cold-blooded killer, and the secret lies in its tail. The tail is the powerhouse of the great white shark and the source of its explosive thrust. It is jam-packed with muscle.
Okay, so you can actually clearly see the differences in muscle types here.
The white muscle is used for incredible bursts of speed and high impact acceleration. But this raw power is not sustainable.
Yeah, the problem with the white muscle is that it lacks the blood supply that the red muscle has got, which is why it's white. And as a result, it can only sustain those bursts of speed for relatively short periods of time.
While the white muscles provide the short turbo boosts, it's these red muscles that drive the shark. They run the entire length of the great white's body and pump the tail like two giant hydraulic pistons. This continuous firing generates enough heat to warm the shark's blood and its internal organs. The tail of the great white acts like a blast furnace. By warming their blood, everything improves. They can digest food quicker. They can swim faster. Their muscles are stronger and more powerful for sudden bursts of speed to ambush prey. But this blast furnace requires fuel, 10 times more than a human. The small bony fish found in warm waters doesn't satisfy the great white's needs. So it exploits its warm blood supply to gain access into the colder waters where all the rich, fatty, high-calorie prey hide. The kind of cold waters found at Seal Island.
All right, so there we go. Once again, poikilotherm, right? He's keeping himself warm. Let's see if we can guess if these are going to be warm-blooded, cold-blooded, poikilotherms, endotherm, ectotherm, all those. So what do you think about a dolphin? That's easy. Mammal. It's a mammal, so therefore it's endothermic, homeotherm, right? Warm-blooded. What about an angelfish? I think we're talking about like little angelfish. You see them? Yeah. Any guesses on that one? I think it's going to be a straight-up ectotherm. Yeah, right. Oh, it does say it's poikilothermic.
Oh.
That's interesting. And then our shark, we know, endothermic, poikilothermic. Well, so see, that's what confuses me. This is what was given to me. I would say ectothermic, poikilothermic. But technically, because it's poikilothermic, it makes it quote-unquote warm-blooded, which is why the German guy kept saying warm-blooded. Do you get it? It's just a technical way of saying that. So that's how our little chart you wrote down earlier fixes that as well. Okay. And remember, this isn't every shark. I probably should have put on there great white shark. Or, you know, not all of them do that. All right. Okay, we're moving on to reproduction strategies. Okay, remember these creatures live in the water. And so there are, just like any living thing, most living things have asexual reproduction and sexual reproduction. Asexual reproduction is reproduction of a single organism or a single cell. Okay, so like a cell in a multi-cellular organism can produce through asexual reproduction a new cell. Okay, so if we're looking at a single-celled organism, something called binary fission, that's what bacteria do. There's one bacteria, they duplicate their DNA or RNA, and they squeeze out two then. So one becomes two. That's kind of what's happening down here. I think we're looking at an amoeba, where it just takes its body, duplicates its nuclear material, and it splits into two identical offspring. So this is a cloning process, okay? So this asexual reproduction is a type of cloning. The offspring look identical to the parent. All right, we have something called runners. and we have something called budding. Budding is going to be the main thing that we're going to be looking at here, because we have creatures out there that will just bud a little baby off of its side. And that's a new baby coming off there. The king of that, of course, would be our hydra. Okay, what we call budding hydra. So here's our hydra. Here's a little bud, and it finally grows out big enough, and it just falls off and becomes a new creature. Okay, the creature is identical to the parent. Okay, so it's a form of binary fission, but it's called budding here. Sexual reproduction involves the union of gametes from two organisms, male and female, genes from the mom, genes from the dad, and that's using specialized cells that are called haploid gametes. Haploid gametes. The word haploid, you should remember from biology 1, means half. So it has half of the genetic material in it. And therefore, that takes half of it and adds it to the half from somewhere else, and we have a new creature. And the process by getting the haploid gamete is a process called meiosis, whereas just getting a cloning type thing is called mitosis. We'll see that here in just a second. Lots of bubbles in here. Okay, and I have a picture that might help you to understand this a little better, which you should also remember from biology 1. All right, so here we have in mitosis, we have a parent cell. They completely replicate into two daughter cells that are identical to the parent cell. That's called mitosis. Okay, that's happening in multicellular organisms. It's happening in our skin right now, and our skin reproduces all the time. It's going through mitosis constantly. That's why skin injuries heal so quickly, because there's lots of mitosis. Where other places the mitosis is very slow, your bones, for instance, therefore it takes longer for a bone to heal after an injury than it does your skin. Now, meiosis, on the other hand, is going to start with one parent cell and end up with kind of two processes of mitosis, and it goes through and ends up with 4 daughter cells, each with only half of the genetic information. So it does not look like the parent cell. Okay, it only looks like half of the parent cell. So asexual reproduction is single organism going through division. Sexual reproduction is 2 organisms sharing genetics. So I take one of those cells and mix it with the gamete from the other sex, and then we have a new offspring. So what about the strategies for marine life? All living organisms develop strategies to create offspring. That's the goal. And in the ocean, we have something known as broadcast spawning. Okay? That releases gametes just into the water. And like they're just broadcast, like you broadcast seed into your garden, right? Other organisms will go and seek out a mate. That would be our mammals. Some of our bigger fish, things like this. But the broadcast spawning we'll see in creatures like, I'll show you a picture here of a giant sponge. A sponge is an animal, and it sends its gametes out into the water and just hopes that one of those gametes meets up with a random gamete from another sponge somewhere. They kind of live in the same vicinity, same neighborhood. And then those will drop down. When we talk about sponges, we'll look at detail of how that happens. So these are both types of sexual reproduction, therefore. A sponge can also bud. Sponges are pretty cool, actually. You can cut them into 15 pieces and have 15 new sponges. We know that about the starfish, right? You cut off its limb and grow a new one. All right, so this is a giant sponge, and you can see spawning into the water around it, hoping that lands near another gamete somewhere. All right. So there are some famous sponge spawning areas that divers go down. Like one night in the whole year does the spawning take place. They go down with their cameras and try to catch it and see. They see something that looks like this. So here's some more broadcast spawning. So we see just these fish are releasing eggs, and then those eggs will just. Oh, that's a mussel like a clam, kind of a thing. And you can see they're releasing their gametes out into the water there. And they're living in a group with other mussels, so they tend to find a mate there. We have mating pairs here. We see our dolphins. Our other mammals, of course, would be mating pairs as well. Other strategies. Eggs are laid. Not all, there are, the mammals, of course, give live birth. That's what makes them mammals. But most of the other creatures lay eggs or hold eggs inside. Okay, so eggs can be laid, they can be laid first and then fertilized afterwards, after they're laid. You can think about fish do that, frogs do that. Also, when they hatch out, once they're fertilized, they can hatch out and have really interesting larval stages. And those larval stages look very different from the adults. Think about a larval stage of an insect, for instance. You know, they look different sometimes than the adult. They change during maturation. And this all becomes very important for the food chain that's taking place in the ocean. Those larvae are very, very good food sources for many creatures in the ocean. So there's usually lots and lots and lots of them. And we'll look at some of these as we come to them when we're talking about the actual organisms, the creatures. Let me tell y'all, we have a lot of dissections in this class, right? I ordered our first semester of dissections in the beginning of August, and they have still not been delivered to me. So, and I contacted them. They said, yeah, we're behind in the you know, I paid for them, but they're behind in the warehouse or something. So hopefully they'll get here in time for us to have our first dissection. Okay, here we're looking at some eggs right there. You can see, what do you think those are? I guess they could be frogs. I thought they were little fish, maybe. I don't know.
They look like little chibi birds, though.
Yeah, I know. And but see, this is their little yolk sac right here. So that's what's feeding them. Yeah. So I was thinking, you know, caviar. Yeah. All right. Okay. Now we're going to get to the naming of creatures. You know, scientists are always naming things, coming up with new words, things like this. Y'all should all remember all of this. Classifying organisms using their genus, species, name. We're going to be remembering how most scientists will use the binomial nomenclature. Okay, so that's their genus, species, name. They're changing things in other ways, like instead of calling them, you know, in certain kingdoms, they might call them domains and things like this. So when you get to college, things might be different because they're kind of sort of wanting to take Linnaeus out of it. So Carolus Linnaeus was the one who set this up with kingdom, phylum, class, order, family, genus, species, and he was a believer. And some scientists want to get rid of that guy, which is sad, you know, because he's a great guy. So kingdom, phylum, class, order, family, genus, species. Kings play chess on fine glass sets. Y'all may have another mnemonic that helps you remember that. If I ask you to tell me this, I'm not asking for kings play chess on fine glass sets. I'm asking for kingdom, phylum, class, order, family, genus, species. Okay? Kingdom, phylum. Did I miss one? Kings play chess on fine glass. There it is. Kingdom, phylum, class, order, family, genus, species. So every creature out there has at least this many names. Okay, so we're going to name one right here. Here's one. So we have, this is a fish in the ocean. It's in kingdom Animalia. Phylum Chordata, that means it has a backbone. Class Osteichthyes, which is fish. Order, all right, Tetradontiformis. That sounds like maybe he has 4 teeth. Tetradonta, right? Family, Ballastadia. Genus, Ballastapus. Species, Undulatus. So those names all refer to the Ballastapus undulatus is the orange-lined triggerfish. It's got all those names. Notice the words are in Latin. And if they're in Latin and you have them printed out, those would be italicized. See, they're italicized. If you're writing them out, you would underline them to show that they're Latin. Also notice our genus is capitalized and our species is not. It's a lowercase letter there. So, Ballastapus undulatus, that is the correct writing of that word right there that is describing this creature. All right, now we're going to do lab time. Okay, let's take a look at this. You should see a difference. I might need to bring it to you.
Hey, what's going on here?
Oh, it's osmosis. Oh, no, we are on osmosis is back.
That's what I have. Osmosis, yeah?
Is that this one or a different one?
Oh no, this is different. This is respiration. We have 2 today, yeah. And so I'm going to end this here.
Do we need to write anything down? It says experiment 2.3 and experiment 2.4 for demonstration of diffusion and demonstration of osmosis.
let me take a look at it. Here, homework for next time. Oh, I turned it off. Module 2, finish it with the study guide. It says there's a summary in the back of the book. Have y'all looked at those summaries back there? You can do that if you like. And then you can either take test 2 before next week or take test 2 after next week. That's up to you. All right?